Hello everyone. This video will focus on the part 2 for the past year NEET questions for principles of inheritance and variation. So let's start. So in a population of 1000 individuals, 360 belong to the genotype AA, 482 capital A small a and the remaining 162 small a. Based on this data, the frequency of allele A in the population is what? It's a very simple question that is based on the Hardy-Weinberg Principle that says that the frequency of two alleles and their square is 1. P and Q are the different alleles, or it is P square plus 2PQ plus Q square is equal to 1. Okay, so what is P square? AA is P square here, and what is it here? The P square is 360 in 1000. So you will divide 360 by 1000, which will be 0.36. So 0.36 is P square, and what you have to find is P, which is under root of 0.36, which is 0.6. So the correct answer would be 0.6. Okay, so according to the Hardy-Weinberg principle, this is the equation. And sometimes you can also do it, the other way, because we know that P plus Q is equal to 1. So Q square is here 160 out of 1000, that is 16 out of 100 under root of 16. And then you can multiply. Sometimes what they can do is they can only give you the frequency of the small gene, and then they ask you that for the allele A. Okay. So then it is important that you must know that P plus Q is 1, and in that case you will use, like, this is a bit longer solution. So this is how it is. You just need to know the Hardy-Weinberg principle, and it's a very simple question. So the answer is C, 0.6. Okay. If the two persons with AB blood group marry and have sufficiently large number of children, these children could be classified as A blood group, AB blood group, B blood group in 1:2:1 ratio. Modern technique of protein electrophoresis reveals presence of both A and B type proteins in AB blood group. This is an example of, it's such a long question, but just what is this example of that? The presence of both A and B type protein in AB blood group. So what is this? Is it incomplete dominance, partial dominance, complete dominance, or codominance? It is codominance, when both A and B is expressed. So ABO blood group system in human begins as an example of codominant, dominant recessive and multiple alleles. Blood groups are controlled by gene 1 located on the 9th chromosome and has 3 multiple alleles, out of which any 2 are found in a person. In codominance, both genes express themselves completely. For example, A and B are both expressed. That is why we have blood group AB. So this is example of codominance, and the correct answer is D. The next question is, which Mendelian idea is depicted by a cross in which the F1 generation resembles both the parents? Is it law of dominance? It is inheritance of one gene, codominance, incomplete dominance. So the F1 generation resembles both the parents, and we just did this question previously. The correct answer here is codominance. In codominance, F1 generation resemble both the parents. For example, blood group inheritance. You have like kind of repeating questions, So it's basically very nice that if you go through all these questions and then you know what areas to focus on. The answer is C. Which one of the following statements is not true? Read the question properly always, okay? It's not true of two genes that show 50% recombination frequency. The genes are tightly linked. The genes show independent assortment. If the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. The genes may be on different chromosomes. So what is not true is that the genes are tightly linked. And why is that? This is because when the genes are tightly linked, there is very less recombination taking place. We saw a similar question in the part 1. So the questions are similar, the way of asking is different. Okay, so the correct answer is A, because this is not true. If there is 50% recombination, the genes are not tightly linked. But the genes show independent assortment. If the genes are present on the same chromosome, they undergo more than one crossover, and the genes may be on different chromosomes. Okay, so tightly linked genes show more linkages than crossover. Genetic variation in a population arises due to mutations only, recombinations only, mutations as well as recombination, reproductive isolation and selection. So the correct answer here is mutations as well as recombination. Okay, previously there was a question where we saw in the part 1 it was recombination, but it is mutation as well, because there is always mutation. You are similar to your parents but yet different. Why? Because there was mutations that took place. Crossing over leads to recombination of genetic material on two chromosomes. Mutation results in alteration of DNA sequences and consequently results in change in genotype and phenotype of an organism. Okay. So in addition to recombination, mutation is another phenomena that leads to variation in DNA. So the correct answer is C, mutations as well as recombination. A certain road accident patient with unknown blood group needs immediate blood transfusion. His one doctor friend at once offers his blood. What was the blood group of the donor? So here you have to really keep in mind that the blood group of the donor should be one that does not elicit an immune response. So what will be the answer? Is it B, AB, O or A? It is blood group O. This is because there is no antigen that is present on blood group O and there will be no immunogenic response. Okay, that is why blood group O acts as a universal donor. So answer is C, blood group O. A test cross is carried out to determine the genotype of a plant at F2, predict whether two traits are linked, assess the number of alleles in a gene, determine whether two species or varieties will breed successfully. So we all know what is the answer. A test cross is carried out to determine the genotype of a plant at F2. Okay, a test cross first introduced by Gregor Mendel is used to determine if an individual exhibiting a dominant trait is homozygous or heterozygous for that trait. It takes place between the F1 generation and the recessive parent. You always cross the F1 generation with the recessive parent to determine the genotype of plant at F2. So the answer is A. F2 generation in a Mendelian cross showed that both genotypic and phenotypic ratios are same, 1:2:1. It represents a case of what? So what in the normal case what happens is the genotypic and phenotypic ratios are different. It is 1:2:1 and 3:1. Okay. But here both the genotypic and phenotypic ratios are 1:1. So what is it? Is it codominance? It is dihybrid cross. Is it monohybrid cross with complete dominance? Is it monohybrid cross with incomplete dominance? It cannot be codominance. It is not a dihybrid cross. It is not a monohybrid cross with complete dominance because the ratio is 1:2:1. It is a monohybrid cross with incomplete dominance. Okay, so incomplete, partial or mosaic dominance is the phenomena where none of the two contrasting alleles or factors is dominant. Okay, the expression of the character in a hybrid or F1 individual is intermediate or a fine mixture of expression of the two factors as found in homozygous state. It is very important. Incomplete or mosaic inheritance is not an example of pre-Mendelian concept of blending inheritance because the parental types reappear in the F2 generation. It is, however, considered by some workers to be an example of quantitative inheritance where only a single gene pair is involved. F2 phenotypic ratio is 1:2:1, similar to the genotypic ratio. So you must remember in case of incomplete dominance, the ratios are both same, in F2, 1:2:1. So the correct answer is D. When two unrelated individuals or lines are crossed, the performance of F1 hybrid is often superior to both the parents. This phenomena is called, it is a very simple question where you need to know the definition and the answer is heterosis. So heterosis or hybrid vigor occurs when two unrelated individuals or lines are crossed. to the performance of F1 hybrid, which is often superior to both its parents. So the answer is A. A person with unknown blood group under the ABO system has suffered much blood loss in an accident and needs immediate blood transfusion. His one friend who has a valid certificate of his own blood group type offers blood donation without delay. What would have been the type of blood group of the donor friend? What would be? We just did a similar question, it should be type O because it has no antigen and therefore no immunogenic response will be there. So even if the person who needs blood is type B, it can take O. Type AB can take O and type A again can take O. You see, there are similar questions repeating over the years. So type O blood group is considered universal donor blood group. It can potentially be transfused to any patient regardless of their blood type. In emergency situation, this type of blood can be given to the patient. So the answer is C, type O. Test cross in plants or in Drosophila involves crossing between two genotypes with recessive traits, between two F1 hybrids, the F1 hybrid with double recessive genotype, between two genotypes with a dominant trait. We just saw this, previously. The answer is the F1 hybrid with a double recessive genotype is what is a test cross. In a test cross, genotype of an organism showing dominant phenotype is determined by crossing it with the homozygous recessive phenotype. So the answer is C. Which one of the following conditions of the zygotic cell will lead to the birth of a normal human female child? 2 X chromosomes, only one Y chromosomes, only one X chromosome, one X and one Y chromosome. So one X and one Y chromosome leads to male. These are abnormal characteristics. So the correct answer is normal human female child will have 2 X chromosomes. 2 X chromosomes would lead to the birth of normal human female child. So the answer is A. Which one of the following cannot be explained on the basis of Mendel's law of dominance? So you should definitely read the question. It is asking about just the law of dominance. Mendel also gave other law, right? The law of independent assortment. So you should definitely know what comes under what. The discrete unit in a controlling particular character is called as a factor. Out of one pair of the factors, one is dominant and the other is recessive. Alleles do not show any blending and both characters recover as such in F2 generation and factors occurs in pair. So what is the question here? Read it properly. Mendel's law of dominance. All these are all fine. All the statements are correct, but what can be explained on the basis of law of dominance? The discrete unit controlling a particular character is factor. Yes. Out of one pair of factor, one is dominant, other is recessive. Yes. And factors occur in pairs. So these can be explained by law of dominance. Alleles do not show any blending and both characters recover as such in F2 cannot be explained by the Mendel's law of dominance. So the correct answer is C. According to Mendel's law of dominance, out of two contrasting allelomorphic factors, only one expresses itself in an individual. The factor that expresses itself is called as dominant, while the other, which has not shown its effect in the heterozygous, is termed as recessive. Option C in the given question cannot be explained on the basis of law of dominance. It can only be explained on the basis of Mendel's law of independent assortment, according to which in a dihybrid cross, the two alleles of each character assort independently of the alleles of the other character and separate at the time of gamete formation. Okay, so it is very important that you know which factor explains what, or which Mendel's law explains what all characteristics. So the correct answer is C. The genotype of a plant showing the dominant phenotype can be determined by a test cross, dihybrid cross, pedigree analysis or back cross. So what is the answer? See, we are seeing similar kinds of question, So the genotype of a plant showing dominant phenotype can be determined by a test cross. So test cross is the cross of an individual with an individual having recessive phenotype. It is used to determine genotype of a plant showing dominant phenotype. That means to determine whether the individual exhibiting dominant characteristics are homozygous dominant or heterozygous dominant. So it is a test cross. So the answer is A. You see, there are similar questions repeating. Select the correct statement from the ones given below with respect to the dihybrid cross. Tightly linked genes on the same chromosome show higher recombination. So you have to find the correct statement. Is this statement correct or not? Tightly linked genes on the same chromosome show higher recombination. Genes far apart on the same chromosome show very few combinations. Genes loosely linked on the same chromosome show similar recombination as tightly linked ones. Tightly linked genes on the same chromosome show very few recombinations. So what is the correct answer here? The correct answer here is D. So the tightly linked genes on the same chromosome show very few recombinations, and we have already discussed it a lot of times in part 1 and part 2 in different kinds of questions. Linkage is an inheritance of genes of same chromosome together and capacity of these genes to retain their parental combination in subsequent generation. The strength of linkage between two genes is inversely proportional to the distance between the two. Okay, this is very important. The strength is inversely proportional to the distance between the two. The two linked genes show higher frequency of recombination if the distance between them is higher and lower frequency if the distance is smaller. Okay, so the correct answer is tightly linked genes on the same chromosomes show very few recombinations. So this was the end of part 2. In subsequent parts, we will see more questions. Thank you for watching.